my talk slides. Anyway, so what I want to do in the wrap up here is convince you that microbes run the planet. So I'm done, right? I used to be a birder, obsessed birder. I get obsessed with things, I guess, here and there. And my dream is to make an equivalent of a field guide that we have for birds and butterflies and bugs and flowers for microbes. And sequencing technology is helping us do this really well. And so what I'm going to do is just take you through some examples of what I call phylogenetic or phylogenomic analysis methods or sort of approaches to understanding microbial diversity using DNA sequencing. And the first one I'm going to talk about is something called phylotyping. And what phylotyping is, it's sort of derived from methods originally pioneered by Norm Pace and colleagues, where you go to an environmental sample, you extract DNA from that sample, you run PCR targeting specific genes, usually ribosomal RNA genes because they have good PCR primers. You sequence some of those ribosomal RNA genes, and then you make a multiple sequence alignment of them to compare them to each other as well as to genes that you've sequenced previously. And then what you do to make it full phylotyping is some type of phylogenetic or comparative analysis. And there are just a few different paths to this. I just thought I would outline a couple of these different paths. You take a multiple sequence alignment. The true phylotyping would take that and build a phylogenetic tree and then try and do something and analyze that phylogenetic tree. For example, you can look in the tree for clusters of sequences from your environmental sample that are really closely related to each other, call them species. Unfortunately, we call them OTUs, or operational taxonomic units, but pretend they're something like a species. Build that tree, compare them to each other, and compare them to organisms that have been characterized and try and make sense out of the environment or the organism. An alternative approach, which is also done, is to take the multiple sequence alignment and just compare the sequences to each other and look at the percent identity of them and cluster them based upon their percent identity, sort of a quick and dirty method, find clusters within that big matrix, distance matrix, and identify operational taxonomic units from those clusters. And then sometimes people will then build an evolutionary tree from those refined operational taxonomic units, so not analyzing all the data, but just representatives of each group. The true obsessed phylogeneticist would build an evolutionary tree of all the sequences and just use that, and never try and assign things to a species or an operational taxonomic unit, but analyze the entire data set. And I'll give you examples in essence of all three of these. So why do we do phylotyping? Well, for a long time, we've been interested in trying to characterize the microbial world around us. And just like with the bird field guide, we would like to have a list of organisms from a particular environment. We would like to know relative abundance of those organisms, some ecological metric, the diversity of different kinds of organisms in an environment. We would like to use phylogeny to characterize the organisms in that environment. We can look at rates of change, groups of organisms that are related to each other. We can look at convergent evolution. There's a lot of diverse purposes. In essence, taking the 150 years of plant and animal phylogenetic approaches to ecology, or just ecology, and applying them to microbes. And the data that we have is primarily dominated by sequence data these days. So what I want to give you a couple of examples of is what's new in phylotyping, especially in the context of new sequencing methods. So one thing that's new is you can still do ribosomal RNA PCR, and lots of people still do, but what we can do is sequence very, 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 very deeply into particular environments and characterize the tail end of the distribution, the relative abundance distribution curve, for example, something called the rare biosphere. Or what people are really doing more of is to process more samples, especially with barcoding methods. We can do time series. We can do spatially diverse sampling. We can do very, very, very fine scale sampling. There's a great paper by Rob Knight and colleagues where they sampled one of his graduate students' faces in 45 different places along his face. This would be unheard of with microbial studies 10 years ago, but sequencing allows us to characterize deeply. I'm just going to give you one example of this. This is something called the Earth Microbiome Project. I'm not actually heavily involved in this. It's run by someone named Jack Gilbert at Argonne National Lab. I'm sort of an advisor to them. And when he first suggested this to me, I thought he was literally insane. He wants to sample the entire planet, all the microbes on the planet. I mean, there are like 800 quadrillion niches out there that we need to sample over time as well. And it just seems completely crazy. But the concept of sampling microbes on the entire planet is actually starting to get feasible at some level. Now, obviously, we're not going to sample literally everything, but Jack Gilbert has shown, for example, in a recent paper where they sampled across the English Channel, that they can not only sample diversity of the microbes in the English Channel, they can use that sampling, those fine-scale sampling, to measure ranges of microorganisms, to predict where microbes are going to be found in environments that have not yet been sampled. That's what we truly need in terms of microbial diversity, not to just study one site, but to be able to predict what we're going to find in other places. Again, just like that field guide to birds, I know within a close approximation what birds I'm going to find around the Claremont Hotel. Pigeons and other things. But you can predict that from a good field guide with good range maps. Just as a thought exercise, I was thinking about this yesterday. So the Mississippi River, 2,320 miles long. Let's say you do 3 samples per site, 1 site per mile. That's 6,960 samples. As a microbial diversity person, that just seems crazy to me. But in retrospect, with a HiSeq 2000 run, if it all works, and you get, say, 6 billion sequence reads, that's 862,000 sequences per sample. That's more than enough. I published my first scientific paper on 1 ribosomal RNA sequence in a sample. So I'll take 862,000 per sample and do some biogeography of the Mississippi River, maybe get Huck Finn to go down it in his raft or something to that effect. We could even say, let's do it every 500 feet. And actually, it's not crazy these days. I mean, the rate-limiting step is actually collecting the samples. The next rate-limiting step is processing the samples. And the final rate-limiting step is bioinformatics. So getting the sequence done is actually perfectly reasonable if you're going to use ribosomal RNA sequencing to characterize samples. 81,000 sequences per sample is actually more than was published in any ribosomal RNA survey until about 2 or 3 years ago. So this is just revolutionizing the approach to microbial diversity on the planet, is the ability to sequence tens of thousands of ribosomal RNA sequences and do phylogenetic typing and phylogenetic ecology across a wide diversity of ecosystems. Now, the other thing which you've heard a little bit about here at this meeting, which is new in phylotyping, is metagenomics. So ribosomal RNA PCR is great. You go to an environment, you PCR amplify ribosomal RNA genes. We know a lot about the diversity of ribosomal RNA genes. We have a big database we can compare to. But they're imperfect for a lot of purposes. So, for example, if you want to study viruses, they don't have ribosomal RNA genes. So if you want to do things about function in organisms, ribosomal RNA doesn't tell us a lot about function. So a lot of people have switched to going after the whole genomes of organisms in an environment. That's called metagenomics. Now, if you did 81,000 sequence reads per sample with metagenomics, you're going to have very low scraping across the diversity of organisms in those samples. So you need more sequences per sample to make sense out of it. Not everybody's going to do as many as Eddie Rubin would do in the sheep rumen, but you could do hundreds of thousands to millions of sequences per sample and characterize a lot of samples with metagenomics. And you can apply the same general approach as done for ribosomal RNA to look at the phylogenetic diversity of organisms from metagenomes. The thing I want to talk about a little bit here is why that's sort of hard with metagenomes as opposed to ribosomal RNA PCR. So the main challenge is that we get fragmentary pieces of data. Even with longer reads that we're getting now, we're still usually getting fragments of particular genes. So if we make a multiple sequence alignment and compare the sequences to each other, sometimes sequence number 3 doesn't overlap with sequence number 1. And how do we make sense out of sequences that don't overlap with each other, especially if they're from different taxa, as well as from a particular sample? So this has been a big challenge in metagenomic analysis, and there have been sort of 3 classes of solution to this challenge. One is to treat each sequence as an island, to analyze each individual sequence in comparison to a reference database, and then analyze the next sequence in comparison to the reference database, and the third sequence in comparison to the reference database. You can consider this sort of like tiling reads against a reference genome. Sort of the same general idea, but now we're analyzing massive collections of sequences from organisms compared to a reference database of a multiple sequence alignment from organisms for which, say, we have the whole complete genome sequence from. Now, this can be very powerful. As we get more and more sequence data, we need automated methods to do this. My lab has built automated methods to analyze this type of data, as have many other labs. We have one tool called Amphora. There are probably 10 or 15 of these tools out there to take each individual sequence on their own. And we prefer to use phylogeny because that adds and allows us to use phylogenetic approaches, the full richness of looking at the tree and the history of genes, as opposed to similarity-based methods like BLAST. So we've done this in a lot of environments, as have lots of other people, this sort of one-off, one sequence against the reference database. But that's not ideal. We want to know how sequence number 1 relates to sequence number 3, to sequence number 11, to sequence number 40. So how are we going to compare them to each other? One thing to do is to sort of cheat, is to look at your multiple sequence alignment and find regions where, some of your environmental sequences overlap with each other. Chop out anything that doesn't carry a lot of sequence in that sort of core region of a particular gene, and just do your phylotyping with that. That's what I did in the Venter Sargasso Sea, sort of one of the first big-scale metagenomic studies to go along with Jill Banfield's study, and came out at about the same time. So we can build phylogenetic trees of all the ribosomal RNAs that have that core region and characterize them just like you would do with ribosomal RNA PCR. The great thing about metagenomics is that it allows you to finally look at other genes to do phylotyping of microbes. So because ribosomal RNA PCR is pretty easy to amplify genes from a diversity of organisms, that's what most people have used when they're using PCR-based approaches. But ribosomal RNA is imperfect for a lot of phylotyping and ecological purposes. For example, the copy number varies enormously between organisms. If you want to estimate relative abundance of an organism from the number of sequences that you get of its ribosomal RNA gene, it's pretty inaccurate if you don't know the copy number of that gene. There are many protein coding genes, however, that have very low variance in copy number between organisms. And we can use those, build phylogenetic trees of those, and make a much more accurate estimate of relative abundance of particular taxa, and use ecological methods that rely upon good estimates of relative abundance to calculate things like beta diversity and other parameters from the ecology of communities. So we've used a diversity of universal protein coding genes to do this, like REC-A and RPOB and HSP70 and elongation factors, and you can sort of do the same type of analysis that you would do with ribosomal RNA with protein coding genes now for the first time. And that, for example, opens up viral diversity studies, because you can pick a phylogenetic marker from a particular class of viruses and look at their phylogenetic diversity just like you would do with cellular organisms. There are also a lot of tools that will help you do this. Many of them are really nice and high throughput and robust. Mother and Chime in particular. You can do this with ribosomal RNA. They're not yet really tuned for doing protein coding sequences per se, but you can use protein coding sequences in them. The holy grail of doing this for analyzing metagenomic sequences is to not throw away any of the data. So even sequences that don't overlap in a core region with the other sequences we would like to use. We would like to analyze everything in our data sets. So take a data set like this, where the full-length things here are from the reference database, the little things are from our environment, and analyze it all. So compare them to each other. Now, how can we do that if this sequence doesn't overlap with that sequence? How can we build an evolutionary tree that includes both of them? Well, it turns out many people have been thinking about this for a long time, in particular, for example, for EST data. People have been wondering, if I have a EST from the left half of a gene and from the right half of a gene, how can I figure out what it is? So there are a lot of methods that people have developed for trying to build some type of evolutionary tree of sequences even when they do not overlap with each other, and then apply, again, the same general phylotyping methods to that type of data set. So we've done this, Tom Sharpton, who's in Katie Pollard's lab, is a collaborator of mine, and he did this for the OTU side of phylogenetic classification. We've done it in my lab for just analyzing all sequences in environmental samples, and you find when you start to analyze all of the data, you can find certainly some very weird things out there, although probably not weirder than the snake viruses that you've heard about here. But for example, if you build evolutionary trees of protein coding genes, you find sequences in a lot of environmental samples that don't group into any of the known major branches in the tree of life. not in archaea, bacteria, or eukaryotes, and not in any obvious viral group. We don't know what these are. They seem to be real sequences that are out there, but the diversity that is out there is enormous. And if we can analyze as much of the metagenomic data as possible, we will start to get some traction on what the true diversity is of life on the planet. So we've developed in my lab, Aaron Darling and Holly Bick and Guillaume Jaspin and others have developed a pipeline for doing this. That is building phylogenetic trees even when sequences don't overlap with each other. It's called PhyloSift. It's linked to a program developed by Eric Mattson called pPlacer that also does this same type of analysis. And now you can analyze, in essence, all of the sequences, even if they don't overlap with each other. You can even push this one step further and analyze sequences from different genes that don't overlap with each other. All these methods rely on having basically a reference alignment that is full length, and you compare your fragmented sequences to each other in the context of a tree of the reference sequences. Steve Kemble, who was in Jessica Green's lab, developed a method for doing this with different genes, concatenating alignments of different genes and then building a tree of all the genomes from environmental samples, even when the sequences come from different genes. This was published in PLOS One last year for doing this. And when you use this, you can now reveal possibly novel things about the ecology of a sample that ribosomal RNA surveys did not reveal. So that's sort of just a big picture zoom through on what phylogenetic typing can do, and I think it's just going to go sort of crazy as we get more and more data coming from environmental samples, because what we haven't done a lot of is rebuild our reference databases with the information coming from environmental samples. But I'm going to switch topics just briefly here and talk about another aspect of phylogenetics and environmental sequencing, which is to make functional predictions for genes. So phylogeny is a very useful tool in a lot of areas of biology, in fact, in all areas of biology. And it's also helpful in making predictions of functions of uncharacterized genes. And I developed a method, this is where my Twitter handle comes from, called Phylogenomics. I kind of regret this because I moan and complain about bad genomics words all the time, and I think I invented one. But the general outline of the method is to take a sequence, build a phylogenetic tree of your sequence compared to homologs of that sequence, overlay experimentally determined functions onto your phylogenetic tree of that gene family, and then predict the function of an uncharacterized gene based upon its position in the tree relative to characterized genes. This is phylotyping, but for protein families and functional prediction as opposed to for species. And this is a very powerful tool in predicting functions for genes. We've used this a lot in metagenomic data, but it doesn't work a lot of the time. It doesn't work a lot of the time because in many cases, genes from environmental sequences have homologs, but none of them have been experimentally characterized. So what we've been trying to do is adapt these methods that are called non-homology functional prediction methods, which make functional predictions for genes based upon some feature other than their sequence similarity to known characterized genes. An example of this is the distribution patterns of genes across species. And we've been trying to use that in the context of metagenomics to make predictions of functions based upon maybe ecological distributions. So this comes to our previous talk, actually. So we've been using these NMF methods as well for analyzing metagenomic data. I'm not going to explain the method either because I can't explain it. But it's non-negative matrix factorization. It's a new sort of highly sort of mathematically favorable method for clustering and looking at correlations among a bunch of objects. And we've been using it with collaborators at a variety of institutions, including Jonathan Dushoff and Josh Weitz, to try and cluster environmental sequences and their gene families and look at whether or not their distribution patterns are related to things like metadata from the environmental samples. And we have a paper in Press in PLOS One that describes doing this for metagenomic data. The last thing I want to talk about is sort of you could call it a philosophical approach to phylogeny and characterizing microbial diversity, which is thinking about what organisms we should study. So this goes back not from environmental studies, but back to organisms that we work on and grow in the lab. So I've been coordinating a project that's sort of ending now at the Joint Genome Institute called the Genomic Encyclopedia of Bacteria and Archaea. And the basic point is that if you look at the phylogenetic diversity of organisms from which genomes are available, it's incredibly biased towards certain taxonomic groups. And we need a much broader representation of phylogenetic diversity in order to make full use of environmental sequence data and make full sense out of environmental sequence data. So this has been this project that started about 4 years ago now at the Joint Genome Institute, involving sort of the full force of everything they can throw in terms of microbial studies at sequencing organisms. It kind of took over their microbial pipeline in a way. And it was an amazing sort of effort by the Joint Genome Institute, and I'm sort of this semi-inside, semi-outside figurehead coordinator for this project, but it's been driven by people largely at the Joint Genome Institute and at the German culture collection, the DSMZ, which grew up organisms that were really weird and hard to grow and then sent DNA to the Joint Genome Institute. And the basic idea is to march through the phylogenetic tree of life, find branches for which there are no genomes, but for which there's a cultured organism, and sequence the genome of that organism. And we've now done about 300 genomes from across the tree of life, actually from bacteria and archaea, but that's the important part of the tree of life. What I want to mention just very briefly at the end here is some of the lessons that have been learned from this project in the context of microbial diversity. So the first lesson is really important for people who may have thought a little bit about microbes. So one reason that many people were skeptical of this Genome Encyclopedia project was there's this issue of lateral gene transfer in bacteria and archaea and in other microorganisms. So it is well known that two closely related microbes might have identical ribosomal RNA sequences, can have very different genome contents. They can, E. coli, K12, and O157H7 differ by up to 30% of their genome in terms of gene content, but their housekeeping genes and their ribosomal RNA genes are basically identical. So there's some amount of gene flow between different lineages in bacteria and archaea that is horizontal. If you draw a vertical evolutionary tree, that's why it's called horizontal gene transfer or lateral gene transfer. And this is a really important issue because it means that if you're interested in characterizing the functional diversity of life, or the diversity of protein families across organisms, if there's been a lot of lateral gene transfer, then walking your way up the ribosomal RNA tree of life will not reveal any novel protein family diversity compared to just randomly sampling organisms, in essence. We did a very simple test of this, which is we asked, how many gene families do you have for different groups of microbes that have different degrees of relatedness to each other in the ribosomal RNA tree? So the first group we looked at was a group of organisms that are in the same species, Streptococcus agalactiae. It's where Hervé Tetelain and other people came up with this term, the pan-genome, which describes within one species you can have a wide diversity of new genes in each organism that you sequence within that species. And if we plot the number of protein families that we see as we add more and more genomes to this data set, we see that there is an increase. Every genome we add adds new protein families when we sequence new members of this species. However, if we do this for a family of bacteria that are more distantly related to each other than within a species, this is the Enterobacteriaceae, we see a higher rate of recovery of protein families per genome. That is, the more distantly related the organisms are on the ribosomal RNA tree, the more likely they are to have different protein families. If we do this for a phylum, it's an even higher rate per genome. If we do this across the bacteria and archaea, it's an even higher rate. So even though there is a lot of lateral gene transfer, and it's an incredibly important process for understanding the biology of organisms, it does not completely swamp out the signal of sort of phylogenetic history that is found in the ribosomal RNA. For you non-microbial people, there's a common term for what we think we're observing here, which is called synapomorphies, shared derived traits like hair in mammals. We think that bacteria and archaea, one lineage of bacteria or archaea, they share derived traits with each other, even with lateral gene transfer occurring. So the second lesson in terms of this Genome Encyclopedia project is that it definitely improved our ability to do phylotyping for metagenomic samples. Having more phylogenetically diverse genomes to compare our metagenomes to helps us classify environmental reads. It helps us do functional predictions from environmental samples and from other sequenced genomes. But it doesn't help a lot. It's a very small benefit in all of these areas. So we analyzed this with the first 56 genomes sequenced in this project. I'm sure it is also true for the first 300 genomes sequenced in the project. So what do we need to do to improve our ability to make functional predictions and improve our ability to do phylotyping? We need to take full advantage of this new sequence data. So one thing we need to do is build new protein families from all of these new genomes in order to help us make functional predictions from environments that are out there. If we don't have homologs in genomes or in our database of protein families, we're not going to even discover that things are proteins in many cases based upon how annotation works. So Tom Sharpton, again, who I mentioned before, and a group of people in my lab and Katie Pollard's lab have compared all the phylogenetic diversity of genomes and built a set of new protein families across all of these genomes. And we have a paper describing this. The data is already out there on a variety of shared sites, so everybody's welcome to use this. We also need to improve our ability to do phylogenetic typing, making use of these new data sets. So we've been going through individual phyla of bacteria and archaea and building phyla-specific phylogenetic markers, as opposed to universal phylogenetic markers found across all bacteria. Now we can build phyla-specific markers, and for example, for some phyla, you can find hundreds of genes that appear to be good phylogenetic markers for those phyla. So that's going to help immensely when you try and classify reads from an environmental sample. Most of the methods that we use require a good reference phylogeny of all the genomes. We and other people are trying to improve the ability to build a reference genome from now, I don't know what the number is now, but with Illumina out there, we probably have 200,000 nearly complete bacterial and archaeal genomes that are available somehow out there. And the last thing I want to mention is that this is the real reason why sequencing from across the diversity of genomes didn't help much in functional predictions and in phylogenetic diversity studies, which is that the phylogenetic diversity of microbes is actually, it's almost unimaginable at some level. This sort of confusing figure shows, the main point of this figure is that if you look at the phylogenetic diversity of known ribosomal RNA sequences, build a tree, count up the branch length in the tree, that's called PD, or phylogenetic diversity, and you sum up the branch length of all the organisms that have been grown in culture in the laboratory. And it represents something like 1/100th of the phylogenetic diversity from known ribosomal RNA sequences. And with new Illumina sequencing and new 454 sequencing and other methods out there, this was based upon a ribosomal RNA database from 4 or 5 years ago. Every environment that people go to, we discover new environmental ribosomal RNAs, but we do not have new cultured organisms to go with them. So the amount of diversity that is out there that we have not sampled at all by looking at cultured organisms and their reference genomes is enormous. And what we need to do is build a reference library of genomes from uncultured organisms to go with a reference library of genomes from cultured organisms. There's now a new project at the Joint Genome Institute run by Tanya Woike that is doing exactly this, what they call GIBA uncultured, to fill in the tree of life and get reference genomes from across the full diversity of bacteria and archaea as opposed to just sort of a subsample of diversity. The final thing we need is, of course, experiments. Just sequencing the crap out of stuff is not going to solve all the world's problems. We need experiments from across the diversity of life. I think I will wrap up there and remind you that of my dream, which is to build this field guide for microbes. And what is a field guide? A field guide basically has a list of organisms, their phylogenetic relationships, some information about the biology of those organisms, and information about the temporal and spatial distribution patterns of those organisms and how to identify them. We are actually getting to the point where you could say we are starting to build a field guide to the microbes, mostly using sequence data. And I will end there and just leave up my acknowledgement slide and thank everyone for listening.
Any questions for Jonathan?
So, with all these new protein families, what about biochemical confirmation of their function? That's going to lag behind and eventually become the bottleneck once you resolve ways of identifying these new families and clustering.
Yeah, so the question is, what is the biochemical function of all these new families? So we're not testing the biochemical function in this case, but that's why I think that we need a functional encyclopedia across phylogeny as well. So not just biochemical function of individual isolated proteins, which is desperately needed, and there is a group, Rich Roberts got a grant to start to work on this, to fill in some biochemistry from across the protein families that are currently annotated as unknown function, and there are many other groups that are working on this. But we also need genetic roles, cellular roles, and so we need experimental studies from across the phylogenetic diversity of organisms to complement in vitro biochemical studies of this protein family diversity. I would note that we submitted thousands of these sequences to the protein structure crystallography initiatives, and many of them have been crystallized. Not that that always tells you the function that they encode, but we need sort of the full force of different approaches to fill in the functional diversity of life as well as the phylogenetic diversity.
So,
Yeah, it works.
so at what point do you stop sequencing? I mean, if there's this continuum of life, or sorry, at what point do you shift resources from simply just trying to get more stuff, knowing that there's just a continuum of all different kinds of variants out there? At what resolution do you say, okay, let's start to shift to functions?
So if you want to shift resources, maybe we should talk about encode first. Oh no, that's my brother. So I mean, I think what we are trying to figure out, so the last slide I deleted before overwhelming you with all the slides that I had, was one showing the rate of recovery of protein families in different phylogenetic groups, and at different at the tips of the tree versus at the base of the tree. So I think we need objective metrics to quantify the benefit that comes from these types of projects, and then decide based upon those objective metrics with a true cost-benefit analysis, which should we put money into. So that's why for our Genome Encyclopedia paper, which came out now sort of 3 years ago, the entire focus of that paper was what are the benefits, or none, of a phylogenetic approach to sequencing, and trying to develop metrics such that we can then say where to stop. And I'll give you an example, one that kind of breaks my heart. So I used to work at Tiger for 8 years, sequencing genomes, and Tiger, and me included, was really big on finishing genomes. Finishing every single last base to some metric of quality at some level, and filling in, ordering all the contigs in every, perfect orientation. Now, finishing is not accelerated at the rate that sequencing has accelerated, and the cost of finishing genomes is pretty high. So in looking at our Genome Encyclopedia project, we basically asked, what would the difference be between just shotgun sequencing some of these genomes and shotgun sequencing and finishing these genomes? There's almost no benefit for the things that we showed were beneficial from finishing. Now, there are other reasons to finish genomes if you're interested in chromosome structure, and there are many reasons to finish genomes. But for the phylogenetic approach to sequencing, no real benefit from finishing. So that immediately is an important cost-benefit analysis. It says if this is the justification for sequencing additional genomes, you shouldn't be putting millions of dollars into finishing them. If there's another justification, fine. But so we're trying to do that. I really believe in you need these objective measures, because I sort of bristle at dumping money into very large-scale projects just because we can do them.
What about expression, shifting to expression sequencing as opposed to just...
Yeah, so the question is, what about shifting to expression sequencing? So we've been talking among many people at the JGI and at LBL and a variety of other places about a phylogenetic functional project, and the first thing to do is expression sequencing. So absolutely. I mean, I think shotgun sequencing the genome, if you're going to do RNA-seq, is a trivial expense. So you do both, but absolutely we need to fill in things like expression sequencing.
Okay, I'm afraid we're going to have to cut this off and take a brief break, but I'd like to thank Jonathan again, Richard, and all of you.